I am pleased to introduce our speaker, Dr. Riley Bernard. Dr. Bernard is an applied wildlife ecologist and assistant professor in the Department of Zoology and Physiology at the University of Wyoming. Her research I heard a phone. Silence. Her research focuses on the ecology and behavior of cryptic species on topics such as foraging, competition, invasive species interactions, the effects of disease on community structure, species susceptibility, and survival. She also uses tools from decision science to ensure the questions she seeks to answer provide the best information for wildlife and natural resource managers to make tractable and robust decisions. Academically, Dr. Bernard has earned a Bachelor of Science from Linfield College, a Master of Science from the University of Hawaii Hilo, and a PhD in Ecology and Evolutionary Biology from the University of Tennessee. Dr. Bernard has conducted research in a number of countries, including Australia, Costa Rica, Cambodia, Puerto Rico, and throughout the United States. Without further ado, please give Dr. Riley a very warm welcome.
Good, thank you. All right, everyone can hear me okay? Perfect. So thank you, Corey, and the Draper Museum for the invitation to talk today. This is very exciting. My first talk here at the Draper Museum, and actually my third time here at the Center of the West. So beautiful area, and Cody's a wonderful town as well. And thank you all for those who are online that are joining. Some of those might be related to me, actually, as well. So the goal of this talk today is to give you a bit of background as to how I got into the world of bats, because it is kind of an unusual taxonomic group to work with, to either convince or to validate your love of bats, and as well as highlight the work of the folks that have been working in my lab conducting research about bats and the wild places they're around the Intermountain and Rocky West. So let's dive in. So I was born and raised in Oregon. That's how I knew that the western meadowlark was actually our state bird. And in Oregon, my parents instilled in my sister and I this love and appreciation of being outside and caring for the environment that we endured. I was introduced to the field, to field work and working with wildlife during a study abroad in Queensland, Australia. And here is where I got a chance to be introduced to really wild spaces like the Australian bush, interesting and very unique creatures like the platypus and the echidna, as well as getting the opportunity, first time, to capture bats using harp traps. And this is a baby bat, very small. I actually got a chance to hang on to one of those for a little bit. So in Australia, fall 2006, as a senior in college, this is where my scientific love of wild places and wild things grew. And as you can see, a certain group of wildlife really intrigued me. So since that time in Australia, like Corey said, I've had the opportunity to study bats throughout much of the United States, Costa Rica, as well as Cambodia. And you can see that these species are also very, very diverse. They have, some have very big ears, this one eats other bats and other vertebrates. We have our smaller insectivorous species, as well as those that have really cool ornaments on their faces. And so beyond having these opportunities to work with bats and to travel to capture them, what exactly is so fascinating about them, even beyond how darn cute they are? So, bats are the only mammal capable of true flight. Flying squirrels fall like Buzz Lightyear with style, but bats are actually going to be flapping like birds. Most species have only 1 to 2 pups per year. They're also born in the breech position and about 1/3 of the weight of a female bat. So that'd be like a 100-pound woman giving birth to a 30-pound child. Yes, exactly. So their population recruitment can be quite slow. So if there are challenges or threats to these bats and you lose a large group in that population, it takes a long time to recoup. Many species are also highly communal. This is a bat box at Guernsey State Park with a bunch of big brown bats and their babies in there. But you'll also find some species are solitary, like our migratory species. And to really put into perspective, bats are not rodents. Rodents do make up a good proportion of all mammalian taxa. However, bats are about represent about a fifth of all mammals globally. And they represent about 1,500 species. We keep finding new ones each year, it seems. And they're found all over the globe, except in Antarctica. So again, they are cute, but not flying rodents. And many species of our bats use echolocation to navigate through the world that they live in. And I'm going to show you a video here that's a thermal imaging camera, and you're going to see some bats that are going to be flying around kind of up in this blue space. And I'm also going to play some sound, and so that is using a detector that converts the echolocation that the bats are using to navigate and find food into something that we can hear. And so with that echolocation, these bats are basically seeing what they're trying to find a lot better. So bats can see, they're not blind, but what they do is they send out those echoes and then it bounces off an object, whether that's a stationary object like a tree or a wind turbine, or something that they want to eat like a moth. And then they get that echo that comes back to them and they can see and actually get that foraging potential. And here what we're looking at is a spectrogram. So basically on this y-axis is frequency. So here we're looking at kilohertz, and then we have time on the y. And each one of these little pulses is what you just heard, the pulses that those bats are emitting and receiving information from. And as bat ecologists, we can actually use these echolocation signals to identify bats, to determine what they're doing when we're recording them, as well as to see if they successfully nabbed a juicy little insect. And what we do is we actually can look at these different phases. So the search phase is kind of a nice slow pulse where they're trying to see if they're getting any echoes out there. And then they hear something, so they're going to start now approaching, and they're going to turn on those high beams a little bit more, basically. And then all of a sudden you hear that buzzing, and this is that terminal buzz phase where we know that they're going to actually be able to capture that target insect. And so from here we can actually be able to ask questions about, are bats utilizing a landscape for foraging, or are they just kind of moving through? So essentially, again, bats can see, but using echolocation is like you turning on the high beams when you're driving down the highway. It helps you see a little bit better, helps them see a little bit better. But do all bats echolocate? Well, that's not the case. So some bats can hear the wing beats of moths or the rustling of insects on the ground, and you might ask how that is. And the way that these bats do this is that these are echolocating bats here. So this is a Rafinesky's big-eared bat and a Townsend's big-eared bat. This is the one that we have here in Wyoming. And if you can see, they have giant ears for one. So it's good to be big-eared every once in a while. And these little ridges that are on their ears, which you can see maybe a little bit better on this Rafinesky's, helps them hear that rustling of insects on leaves. And so they can actually kind of do a little bit of a sneak attack on their insects, because some insects have evolved the capability to hear bat echolocation. So now they've evolved kind of an evolutionary, co-evolutionary arms race to be better at identifying where their food is without having to kind of alert the prey. And of course, some bats don't need echolocation at all because they're fruit eaters, and we tend to see that more on flying foxes, like the really big 6-foot wings bats that we see in Australia and Southeast Asia. So bats also provide a lot of key ecosystem services in every place that they live. And this is a lot of information here, but some of the main takeaways are that bats are very important. They are important for pollinating more than 500 different species of plants, because while they might not be, going for the pollen themselves, they get covered with pollen when they are going after that sweet nectar. Same here. Who likes tequila or margaritas in the audience? Okay, there's some folks. You love bats then. Bats are some of the primary pollinators for agave. If you don't like tequila, that's okay. You can still be a bat advocate. It's fine. We also have these bats that eat blood. And while that might be scary, there's only 3 species in Central and South America. And what's really cool about these species is that they have anticoagulant and antiseptic properties in their saliva, and that information has been useful for heart medication that we use today. Synthetic heart medication that we're trying to create is not nearly as good as the information that we're getting from bat saliva here. There's also, a range in size from 6 inches to 6 feet, so we have kind of the full gamut here of what our bats can do. But ultimately, here in the United States and in North America, we have insectivorous bats, and they save us billions of dollars every year in cost savings so that we're not having to put pesticides on our agro-ecosystems. And to put that into perspective, here is a large graph broken down by county from Boyles et al. In a science article from 2011. And what we can see is that the areas in the red save billions of dollars. And again, that's just by having bats on the landscape and preventing farmers from having to invest in pesticides. And as an example, a single colony of 150 big brown bats in Indiana has been estimated to eat nearly 1.3 million pests per year. And that's contributing to the disruption in population cycles of those agricultural pests. And that's really important. What's also really cool is in this graph, we're looking at damage that's done by insects. And we have a control, and this control is where bats are accessing those corn agroecosystems. And then exclosure here in the blue is actually highlighting where bats were prevented from actually getting at the corn or near the corn. And so what we're seeing here is that when you have bats that are able to access closer to corn, you see a reduction in pest larvae per plant. You see a reduction in the damaged kernels per ear. You see a reduction in the proportion of ears with fungus over time. And you also see that there's less damage to those leaves. And so bats are directly consuming those insects as well as scaring away insects in this kind of landscape of fear. And it's the same as having bat boxes around your house or bats in your backyard. Those bats are going to probably deter those mosquitoes and pests away from your yard, even if they're not directly consuming them. So I hope now that I've convinced you that bats are pretty darn important in general. And now we're going to dig in a little bit more about how we're digging into our bats and the ecology of bats here in Wyoming. So my lab, as Corey stated, is an applied research lab, and our goals are to minimize information gaps that are related to conservation and management of cryptic species. And so ultimately, we want to help increase the capacity for science-driven management. And what that means is that my students and I go to the managers, so whether they're Fish and Wildlife Service, Forest Service, Wyoming Game and Fish, we ask them what they need to know to try to help better improve conservation outcomes. So while my name might be on the lab logo, I wouldn't be anywhere if it weren't for my amazing students and technicians that work in my lab every day for the last 4 years that I've been here in Wyoming. And so they're a huge testament for how much work we've done and how awesome the crews are. So these are just a smattering of the students and technicians that have been in my lab. So in my lab, we use a variety of tools to answer a broad range of applied ecological questions that are related to bats, from capturing and tagging and tracking bats. So here we actually have mist nets, just like birders do, or we have mist nets here to capture bats at night. When we capture them, we might put on little radio tags here, and then we also give them a little bit of bling, which is going to be a unique number for identification for later. We might also be collecting a variety of different samples, biologic samples from these bats, from blood to guano. So this is a guano rainbow here, to also environmental samples for trying to estimate disease presence in the habitats and roosts that they thrive. And we also will put out those bat detectors to help us have long-term data collection to see what kind of species are using a habitat or a landscape, as well as how often they are using it, and to get at that foraging question too, like we looked at before. So in my lab, we are looking at how major conservation and management challenges may affect the long-term persistence of bat populations here in the Rocky Mountain and Intermountain West. And globally, there are a number of different threats to bats, but in North America, we really see kind of these main ones here on the screen. So habitat alteration from removal of habitat or roosts, infrastructure build-out such as roads, which leads to increased traffic volumes. And we're seeing that right now in places like the Shirley Basin and other areas where we have wind farm development, which helps offset climate change outcomes, but it does create more roads and more traffic on those roads, which could disrupt migrations for both ungulates as well as that of our bats. We also see things like disease as well. And so for the rest of this talk, I'm going to focus on what questions students in my lab are attempting to answer based on these pervasive threats that we're encountering here in Wyoming and in places like South Dakota, where another research project is occurring. So the first really big one is disease, and specifically white nose syndrome that's caused by Pseudogymnoascus destructans. I'm going to refer to that as PD from here on out. So white nose syndrome was introduced in North America in Howe's Caverns, so X marks the spot here, in 2006. And what happened is that these cavers came into this site that is a tourist destination as well, and just found piles of dead bats everywhere. And those that were still alive and clinging on to the side of the caves had this white fuzzy growth on their nose, wings, ears, and their tail membranes. And this is unusual. Bats do die during winter. It's an energetically costly period of time. So if bats don't migrate, they're going to hibernate in a place like a cave that's thermally stable, But something like this is very unusual. And so we started seeing that something was happening that is kind of out of the ordinary and leading to some massive declines of our common bat species. So the fungus basically takes advantage of its host during that most energetically stressful period of a bat's life during hibernation. So basically bats will go into hibernation around September, October, and they will reduce their body temperature to kind of some still animation. They'll arouse out of torpor, which is a stasis within hibernation, about every 2 weeks when they're healthy. And they do have kind of a set number of arousals so that they don't starve in the middle of winter, just like bears when they get ready for hibernation as well. They fatten up in the fall to survive winter and hibernation. And then they emerge around March, April, May to go on to migrate to their summer seasonal roosts. So during this period is when white nose syndrome takes advantage of them. And what happens is it starts infiltrating that live mammalian tissue and can lead to major necroses like we see here. And we also see that bats will start exhibiting sickness behavior like our dogs or cats do when they don't feel well. So they'll move to a different location, either within the cave or outside of the cave. And that leads to things like daytime flights or freezing outside the cave. And so since this introduction, we've lost over 6 million bats due to white nose syndrome. And so a future without bats is actually quite scary. Back in 2006 to 2010, we started realizing that we didn't know enough about bats. And so because of that oh no moment, bat biologists started digging deeper into the ecology of bats and found a lot of unknowns and outcomes that kind of countered the status quo. Hibernation behavior varies across species. We do see that bats are foraging during winter in areas like, say, the southeast, where winter is not quite as harsh as it is here in Wyoming. We have also found that bats behave differently in the east than they do here in the west. Even the same species, like a little brown bat that has a continental-wide distribution, behaves differently latitudinally as well as longitudinally. So a lot of fantastic research has been conducted in the last 18 years since white nose syndrome introduction. However, a lot of what we have learned thus far has been really focused on the ecology of bats in the east and it does not translate fully to our western bat species. So that's where my lab kind of comes in. So here's our 18 bat species that we have here in Wyoming. All of our bat species are susceptible to the threats I mentioned earlier. How many of these 18 are susceptible to white nose syndrome? Shout out some numbers. How many? 50%. 50%? All of them? 16? Yeah, you're close. 11. So 11 of our bat species are susceptible to white nose syndrome, meaning that they get the fungus infiltrates their tissue, and they can have deleterious outcomes, i.e. death from disease. Those that are not highlighted in red are species that are either migratory, like our red bats, hoary bats, and silver-haired bats. Our big-eared bats also do not seem to be susceptible. They can have the fungus on them, they can move the fungus, but we're not seeing disease. And then here, this is a Brazilian free-tailed bat, which we do have them down in the southeastern portion of the state. They don't hibernate, at least traditionally. So yeah, 11 of our 18 species are susceptible. However, like in the East, we're seeing that not all of these species are equally as susceptible or experiencing the same kind of outcomes from PD and white nose syndrome. And so the next few slides, I'm going to highlight some of the work that students and technicians in my lab have been doing to try to fill numerous gaps in knowledge related to the ecology and behavior of western bat species now facing this novel challenge from white nose syndrome. So the first project that we really got started in our lab was working on Camp Guernsey Joint Training Center, and we've been working here since 2021. And the habitat in this region is very similar to that in the Black Hills of Wyoming and South Dakota, as well as eastern Montana, where there are nice pockets of ponderosa pine, juniper, and plenty of sagebrush, of course. There's also access to permanent bodies of water as well as ephemeral bodies of water, which is where we tend to be most successful in capturing bats. This region was also determined to be positive for the fungus in this first summer that we were working there. So we were swabbing bats to see if the fungus was there, and it was. So the purpose of our project here, in collaboration with the Wyoming Military Department and U.S. Fish and Wildlife Service, was to see if this area had 3 species of greatest conservation need, and those that are now petitioned or uplisted for endangered species status nationwide due to white nose syndrome. So that's the northern long-eared bat that we can see here, the tricolored bat, and the little brown bat. So to answer these questions related to presence and habitat use, we captured bats on the landscape and placed those small little radio tags on them to try to track them to the roosts that they use during the day. And over the 3 years we worked on this project, we were able to track about a dozen individuals representing 3 species: little brown bats, western small-footed bats, and long-eared bats. So only one of our target species. And what we were seeing is that each of these 3 species that we did track were selecting roosts that were more often in rock cracks and crevices than they were in buildings and trees, which is something that occurs on the eastern side of the range, or eastern side of the United States. So we're finding some pretty interesting things. However, what we're looking at here is a graph of the total captures of bats over these 3 years. So we have total captures on the y-axis going from 0 to about 150. And then we have our different species here. So I have a 4-letter acronym for each of these species, but I have their photos, their mugshots here to help you navigate through. And so what we were finding is that over time, we're seeing changes in the community composition, even just in raw tallies. So we were able to capture our big brown bats and our western small-footed bats with a lot of regularity. We captured them pretty equally across all 3 years. And what's interesting is that our western small-footed bat, who is associated with white nose syndrome, is not experiencing decline nearly as much as our other Myotis species here. And so some of these species we captured more in the first 2 years of our study, and as white nose syndrome has progressed, we haven't captured them since. And this is that long-legged bat that we have not captured since. We are also seeing that we're consistently capturing our migratory species, and our big-eared bats. So again, those species that are not presumed to be susceptible to disease. So we're seeing a decline in once common species, including those myotis, which we assumed were going to be susceptible. However, we didn't know how bad it could be in this group of in eastern Wyoming. We have not captured 2 of our target species, so our northern long-eared bats or our tricolored bats, but we have had our detectors out there and have recorded suspect calls of those, and I have some students working on trying to vet those calls to see if they're true detections. Our myotis, our little brown bats, were captured and tracked to rocks, trees, and buildings. Funny anecdote. Last year, my student, Julia, was capturing these bats, and her technicians tagged a little brown bat. Then they were trying to track it the next day, and the technician was put out 3 miles out on the military base, and the signal kept going towards town, towards Guernsey. So she's hiking on the highway, still signal there. She's like, oh my gosh, if someone has a bat in their pocket, I swear. And so she made it all the way back to the military base and went around their trailer they were staying in. The bat thankfully was not in a pocket, and the transmitter was not in a pocket. The bat had gone to, this is a female bat, had gone to the male's shower room. And there's a roost, a maternity roost in the male shower room on base. So they captured her 3 or 4 miles away, and she was flying that far to actually go back and roost at night, which is pretty funny. But the calls I got that night of, oh my gosh, we think a bat's lost, it's pretty funny. So we have some next steps. With this 3 years of data collection, we're now working on creating what's called a habitat suitability model. So even though we didn't capture northern long-eared bats in this area, we're actually trying to see, is there any habitat at all that could be useful for that bat on those military bases? And that's going to be useful for Wyoming Military Department as well as Fish and Wildlife Service to ensure that if they need to protect habitat as this bat becomes more rare, then they have that knowledge to understand that and to be able to do so. And then we're also monitoring known hibernacula and maternity roosts. The second white-nosed project that we're working on is in the South Dakota Black Hills, and this is being led by my PhD student Renee Lyle. And the goals of Renee's research is to really dig into how that bat community has changed as white-nose syndrome has become established in the region. And here, white-nose has been present since 2017. So she wants to see, what's going on with those bats, as well as what is the health of bats that are actually surviving. So to answer these questions related to estimating the changes in community composition, bat host health, as well as diet, Renee and her team of technicians are capturing those bats. They're putting out detectors as well. And they're doing this on national park lands, including Mount Rushmore, Jewel Cave, and Wind Cave. And while netting for bats is exciting enough, she's had quite the experience with bison interference. So I didn't get a chance to put the video in here, but this year she's had bison come and knock down her nets, run over her nets, run over her acoustic detectors. It's always wild when you're working in the national park, but it's been pretty fun. And so what we can see here is that what the technicians are doing is that they're swabbing the bats for presence of PD. They're looking at wing damage indices, and again, they're collecting as much information as they can, including guano is gold in the bat world. So guano for diet analysis as well as internal parasite analysis. She's collecting external parasites as well. And so we're trying to get a good baseline, holistic view of what these bats are doing and how they're surviving. So looking at this graphic, it kind of builds into this assumption that we're seeing some pretty dramatic declines in our species. So we had, was a team out there led by Dr. Jesse Alston in 2017 through 2019. And so he was doing research in Jewel Cave, and we've been able to actually look at his data and compare it to Renee's first year of study. She's still working on the analysis for her last 2. And so what we see here is that our white-nose syndrome sensitive species, again, those myotis species, are experiencing significant declines. And so in 2022, Renee captured maybe 1 individual of each of these, no little brown bats. So we're seeing massive declines in this group. We're seeing changes, though, in our non-susceptible species. So our big brown bats and our migratory species, and again, that western small-footed bat, we're capturing them more frequently. So we're seeing a change in that community composition over time with as white nose syndrome sensitive species are declining, we're seeing an increase in other species. She's also looked at capture rates between her first 2 seasons here, and anything below this line is bad news for bats. So what this highlights is that across these species that she's captured, anything that goes negative means that she's seeing a decline in overall capture rate. So now we're seeing that she's even seeing declines in our once common species, including our migratory bats. And so something else is happening as well. These are not susceptible to white nose syndrome, but something's going on, whether that's habitat decline and destruction, wind energy development. Our migratory species get hit pretty hard, literally and figuratively, by wind development. One bonus is that she captured 1 individual in this species, our long-legged bat, so that was a 100% increase. So I guess there's some bonuses there, huh? But she's been in the lab too, extracting DNA from guano to investigate what these bats are eating, as well as to determine if they're dealing with high parasitic loads, because that could be another nail in the coffin for some of our bats. Those that are surviving, are they heavily parasitized, and is that leading to poor outcomes? Noah Yazawa, undergrad in my lab, has been really excited when I proposed, hey, do you want to look at a bunch of parasites under a microscope? So he said yes, and so he's been categorizing, identifying what parasites we're getting on the outside of bats, and we're still kind of confused about what this one might be. We still don't know, but Noah's been in the lab looking at these IDs and trying to determine if some species of bats are, have host specificity with their parasites. And in short, yes, they are. So this is looking at parasites that were collected from both the Guernsey and the South Dakota projects, and we're seeing that for the most part, like our big brown bats and our Townsend's billiard bats are heavily parasitized by 1 species, maybe 2 total. And so this is kind of funny. So the Coronarhinus townsendii, they have their own species of parasite. Isn't that cute? So only theirs. And then we have soft ticks and bat bugs and all sorts of really fun, gross little critters as well. But these ectoparasites can also lead the way to helping us understand the health and welfare of our bats in this system. So some next steps for Verne's project is diving into the sequencing results to determine exactly what the parasites and the parasitic load of those internal critters could be, and then looking at her insect prey. We have all the data, it's just now she has to go through the bioinformatics, so it's a fun coding experience for her next. And these results are going to help determine how white nose syndrome is affecting populations in a more nuanced way. So are we starting to see that competitive release where we have a decline in our white nose sensitive species? Do we see an increase in those that are maybe opening up niches or prey availability for ones that are surviving? We might also see that this could be additive or subsequent infections that lead to poor outcomes. And from this work, we've actually been recently funded by the Bureau of Land Management up in the northern Black Hills to work on some more of this research up there to get at some of these species. And a cool thing, this is the first year that we were out there, we captured a couple northern long-eared bats. So there's some still persisting in this region. So moving on from white nose syndrome, the next group of threats to bats that we're attempting to quantify is that habitat destruction and infrastructure build out. And at the end of the day, you might think this is always going to be bad news, but it might not be the case. We have some species of bats that are considered synanthropic, meaning that they live in and around us with our buildings and our structures. And so bats live in many different places. We have our natural habitat like root balls and caves, talus. We have rock cracks, as well as places like our snags. But those synanthropic bats are going to be ones that use bat boxes, culverts, bridges, buildings, and mines. And so a project that we've been working on, that Logan Detweiler, my first graduate student to fledge the roost, he is now an instructor of biology at Casper College. He was looking at bridge use by bats. And over the 2 years of his project, he surveyed over 500 Wyoming Department of Transportation bridges across 19 counties looking for bat use. And when we say bat use, it's not just physical presence of bats, but also looking at things like urine staining and guano. And if you have guano on that vertical surface, you know it's bat, not rat or mouse or anything like that. So he found, even these pictures alone, that bats are using bridges with high frequency. Over 73% of bridges that he surveyed are being used by bats, with about 359 of those sites detected that had at least sign, and we had about 7 day and 6 maternity roosts, meaning we had cute little pictures of mama bats with their babies, as well as a bunch of adults hanging out as well. This is all guano, so you can see that this is a highly used site, and then you have urine staining up here as well. So this is looking at our map of all the regions that, or bridge sites that Logan surveyed. And the main takeaway here is that the pink sites are those that had bats or bat sign, and then the blue are ones that he didn't find bat use at all. And so while we're finding that bats at bridges was really exciting, one question that we wanted to tackle was, what is it about the bridges that make them more attractive for bats? And so the bridge characteristics are going to be best explained the presence of bats, and that can be bridge design, the feature crossed, and even the abutment height. And when I talk about abutment, it's kind of this portion here that's where the road itself meets the edge of that, that stanchion. So presence of roosts was about 2 times more likely at girder bridges and 1.4 times more likely at T-beam. They really didn't use any of those slab bridges at all. Bats want to find things like this that are going to be able to cling on, or areas that are in between here, and slab bridges just lack those features. So they're just kind of like a wall like this. It's not very useful for the bats to grip onto. We also found that roost presence was going to be about 1.5 times more likely at bridges that cross bodies of water, whether they're ephemeral or not. And roost height or abutment height was very, very important. So 3 times more likely to find bats on bridges that had really tall abutments. And that's because bats like to roost at the very top up here. And while they're really good at flying, they like to fall first before they get loft. That's why if you ever put a bat box up, you want to be 10 to 13 feet up in the air, because if they'll just fall. They're not good at taking off, and that's okay. But what's really cool is that while we were looking for day use of bridges, we threw up some cameras just to see how bats are using the bridges at night. And in this case, this is a bridge outside of Casper, Wyoming, and we found bats were down roosting in this cement area down here, but then during nighttime, they were really active all over the place, all over this metal. And they're cleaning, they're digesting, they're talking to each other, they're mating, they're doing all sorts of stuff. So bats use bridges in a multitude of ways. What's also really cool is, like I told you, guano is gold. So we were able to collect guano from about 320 of our bridges, and we were able to use that to identify what bat species were present. So here what we're looking at is species on the x-axis and the total bridges with bats on the y. And you can see that little brown bats are found 100% used. Every single bridge that we sampled for guano had little brown bats present. And then we had our western small-footed bat that's also present in high rates. And we found a total of over 9 species of bats using these bridges. And so this is really important because, like I told you at the top of the story here, little brown bats are highly susceptible to white nose syndrome, and so this could be providing an area for bats to survive. Like maybe this is a place that we can have some sort of mitigation, clean those roosts, make sure that they don't have any white nose or PD anywhere, and they might be able to survive more readily. We also found that, again, this is our map of Wyoming and all the sites that we collected guano at. And those red blobs here are all bridges that had more than 3 bat species roosting at them. So we're finding a lot of really cool information that bridges aren't just a single species roost, they could be using them by many different species. Our 18 species, again, a reminder, and the cool thing with our bridge guano is that we identified a whole new bat species to Wyoming. You hear it first, heard it first. So this is the cave myotis. They're way too far north here. We found them at a bridge that was in Green River, And so we're going to be doing a bit more investigation here. So this gets us to 19 bats in Wyoming. And I have to give a shout out to Ellen Whittle and the Wyoming Natural Diversity Database because they got the 20th species, the canyon bat, last year as well. So we have 20 species. So I kind of lied to you at the beginning. We didn't have 18, but I had to make it fun. So our next steps here are, again, creating a predictive model using all that data that Logan collected to see, because he surveyed 500 bridges. There are almost 3,000 in the state by YDOT alone. So we want to do a predictive model to see what other sites in the state could have bat use and go target those for surveillance. And ultimately, we want to use that to help investigate the ecology of bats using bridges. Specifically, why are some bridges used as maternity roosts versus other? And then when do these bats use them during the summer? And then looking at what factors lead to differential use. So the final little project I want to highlight with my graduate students is looking at kind of the difference of climate change and wind energy development. And specifically, this is looking at Julia Yearout's work looking at Coryneritis townsendii, or big-eared bat, behavior and conservation. And so her research is focused on understanding how best to conserve and monitor these species. And specifically, she's looking at historical use of subterranean sites over time, as well as trying to get a finger on the pulse of how these bats respond to disturbance and local change around the roost sites. In the literature, it's been proposed that this species does not move more than 25 miles between their seasonal roosts. And so we want to see if that's true here, especially as we see that infrastructure build out occurring in places like the Shirley Basin and other regions in Wyoming. So again, her crew works by capturing bats and tagging them, as well as putting out game cameras to look at behavior of bats in these caves, microclimate buttons, and then we have a series of game cameras and acoustic detectors out as well. Her students, or her technicians, are also going into caves to count these bats and to collect information related to differential use as myota species are changing from being more present to not. So in this next video, it's like the first little bit of information we have, because Julia just started her field season, and you're going to hear a loud kind of a dong, something happening at the very beginning. That is a juvenile Townsend's big-eared bat hitting the cave gate, because they don't know how to fly very well at first. Training wheels are still on, but it's pretty funny. That's the bat hitting the cave gate. And then it proceeds to go lay down for a little bit on the wall. You'd be like, oh my gosh, I'm so embarrassed. I hope no one saw this. And then the bat eventually flies out. But this is the kind of the first little bit of information we're getting from Julia's project. So she captured and tracked 9 Townsend's big-eared bats and found that when those females are reproductively active, they come back to the roost sites pretty regularly. But males and non-reproductive females, they tend to switch roosts. Those newly volant bats, not great at navigating cave gates. And her next steps are starting year 2 of the study. So she's going to start, her crew starts in 2 weeks. And then from there, with the data she gets, she's going to be creating models to compare historical and currently used hibernacula to help game and fish better manage and survey their species, as well as to assess connectivity between those seasonal roosts. So as stated at the start of this talk, bats are highly important to the ecosystems that we live in and appreciate. And we also discussed how they're susceptible to numerous threats. And so one important factor is that we can use bats to assess how well an ecosystem is functioning. And so a common way to track this ecosystem decline is through that of bioindicators. And so bioindicators are organisms that react to the quality or characteristics of the environment and their changes. And they're vitally important to track that environmental alterations to be able to take action to try to mitigate those deleterious effects. And so recent studies have actually found that bats are really good as bioindicators. And they can be great bioindicators for things like river systems, agroecosystems, urbanization, bioaccumulation, so on and so forth. However, to know if they're a good measure here in the US, we actually have to collect some baseline information in order to help make those estimates. So freshwater ecosystems cover about less than 1% of the world's surface, but contain 6 to 10% of all species and 1/3 of all vertebrate species worldwide. And that just demonstrates how much of a hotspot or biodiversity hotspot these systems are. And so while they are important habitats for a wide variety of range of aquatic and terrestrial systems, they're some of the most highly degraded systems and habitats here on Earth. And here's just looking at the Colorado River between 1938 and 2005. And you can just see how much things have changed based on the changes in flood flows from damming up and downstream. So I was able to join after the fun, but join the data analysis side of things on this project. And in 2018, the Sesquicentennial Colorado River Exploring Expedition, or SCREE, floated 1,000 miles from Flaming Gorge on the Green to Lake Mead on the Colorado. So 1,000 miles over 70 days. And they just happened to have a bat detector and threw it up every single night. And it was set to record from dusk to dawn, and the placement was very important between the latrine and base camp because they didn't want to forget the bat detector. So every single night they put out this bat detector, and so we had 70 nights across 1,000 miles to see what the baseline information was in this river ecosystem 150 years after Powell had gone through and first started kind of measuring everything out. So the biggest takeaway here is that there's a lot of different species that were found on this entire river trip. There are 19 species, and we recorded bats on every single day, ranging from about 1 call to 3,000 calls. And as you can see, there's a lot of different call files, almost 60,000, with some bat species, like our migratory species, and those that are a little bit more rare here in Wyoming, very dominant on the platter here. This is every single site that we went through, and I have highlighted the physiographic sections here. And so physiographic sections kind of highlight the different ecosystems that are there. They might seem very similar to us by the naked eye, but we're seeing that the bats are actually, the bat community is actually different at these areas. So here the main takeaway from this NMDS plot is to see that our Grand Canyon lands bat community is distinctly different than our Grand Canyons, which is different from our Uinta Basin in Wyoming. So even though we might not be able to detect that difference, the bat community is different. We also found that bats have different activity levels on different types of water systems. So we have Still, which is looking at more of a reservoir type ecosystem. Riffle is more of our rapids, and then Glide is the nice party boat type of slow moving river. And what we find here is looking at number of bat passes on the Y and then the different river types here is that bat activity tends to be more active on Glide and Riffle, and we have less bats detected in these reservoir-like sections, which is kind of counter to what I was thinking. I predicted it'd be opposite. And so what could be happening is that in these still reservoir-like systems, it's kind of just a homogeneous landscape. Whereas if we're in the Glide and Riffle sections, we have a little bit more intact riparian systems that are a little bit more attractive to bats and the insects that they prey upon. So what does this all mean? I'm going to wrap up here. Ultimately, we cannot manage all bat species the same, nor can we manage all populations of the same species the same. Like I said, little brown bats have a continental-wide distribution. They use caves and mines and trees in the east and rocks here in the west. So that means that the information that we gather here on once common species is really helpful in trying to inform place-based management in the right context. But the variety of threats are real. Management decisions are challenging due to uncertainty in the likelihood of a landscape where a species will be affected in one location versus another. And so we need to think about, for instance, how to make our western landscapes fire-wise in the face of climate change. And these are just pictures looking at an unmanaged ponderosa pine site, and then that is managed to its more natural state in Miles City, Montana, where the BLM is really working on this to improve the integrity of these ecosystems. And in point in case here, I told you about the Guernsey project that we have. There's an actual fire occurring right now over there. It's near some of our field sites and some of our maternity roosts. And so again, in this changing climate, we need to be able to be robust and protective of our bat species. And if that means we need to change the way that habitat is done, I'm here to help determine how bats react to that. So by digging into the ecology, we can get that baseline information. Hope is not lost though. Through collaborative efforts of my lab and the various state and federal agencies we work with, we're filling in major knowledge gaps that are already helping natural resource managers effectively conserve a species here in the West. We still have a lot of work to do, but students and technicians in my lab are top notch, so stay tuned for the future to see what our research findings are coming out soon. Thank you very much for your time and attention today, and I'm happy to take any questions.
Thank you, Dr. Bernard. And if we have questions, I will run a mic around to folks so that people online can also hear those words. I'm just going to turn on the lights here so we can see everyone's faces.
Three short questions. One with your bridges. Do you get competition with swallow nesting on bridges?
Yes. So the nice part is that most of the swallow nests are not in the same places that the bats are at. So that's a good thing. So it kind of diminishes some competition. However, there is evidence that certain species like pallid bats will use old cliff swallow nests. And so that's been documented in other regions. However, we have still seen that high swallow activity does not necessarily deter bat use. However, Logan would go out and he'd survey 1 day at a time, and so we have kind of one time point for yes, no, bats are present. And our future studies, we're hoping to see if there's high swallow use, do we see a decrease in the amount of bats that are using that, or they're fine as is. So it's kind of a mixed question. We're not sure, but they do use some of those cliff swallow nests too.
Number two is, have you discussed this with YDOT in terms of maybe improving bat habitat?
Yes, YDOT is very interested in trying to make sure that they do not disrupt bats without having timing stipulations. So bats are using these bridges at the same exact time that construction season happens here in Wyoming, i.e. like 3 months out of the year. So they're not, because it's human health and safety, they have to get that work done. However, they want to make sure that they're not affecting the bat population. So we have some ideas in the work to try to see, is there a way that we can maybe temporarily exclude them from a site and put up some bat boxes so that we're not completely eradicating them from this area, and then they'll be able to utilize that again. There's also some sites down in the Uinta County area that have bat boxes on the bridges themselves to try to enhance habitat. So we've also found PD, the fungus that causes white nose syndrome, on bridge roosts, and so we're trying to talk with YDOT to see, is there a way that we or someone can go in and actually clean that site, so when bats do come in from hibernation, they're entering a clean site and not getting reintroduced by the fungus, because that could be a way that we're transmitting disease to new locations as well.
Is there a way you could improve the nesting?
Yeah,
by spraying some type of material that has some grip to it?
It could be, yeah. It does seem that, I mean, most bridge types, those girder and T-beams, they tend to be pretty happy using those areas as is, or if there's a little bit of cracks and crevices in there. The slab bridges don't tend to be over bodies of water, so it might be more of a ecological trap if we brought bats there, because they have to fly farther to get to foraging areas. But it could be where we enhance little grippies on the under part to make sure that they do stay in certain areas or have a higher likelihood of use. Yeah, that's a possibility for sure.
Okay.
So leading into one of the points you just mentioned, one of the questions I have is, how far might bats travel in a given night for foraging habitat? And I'm sure that depends on the quality of habitat they're in, but is there any literature that you're familiar with, or research that maybe you or your lab has done to look at that?
Yes. So there are the Leptonicterus species, so those long-nosed bats in Mexico, they will fly 80 to 100 miles in a night to go forage, because that agave in Texas is just really good, apparently. Whereas we also have seen tricolored bats in Tennessee that are roosting on lake landscapes, will travel 11 to 20 kilometers across the lake to habitat patches. So whatever habitat patch they're roosting in is not necessarily where they want to eat. They also have shorter movements too. And that's a question that we haven't really answered here in the West. However, I'm glad you asked this question, because we just got funding to work with the Miles City Bureau of BLM, and we're going to get a drone to track bats at night to get at that activity. So we're actually going to be able to have drones track them at their foraging areas to see exactly how far they are foraging from a roost site to a foraging site, because that's another question. If we want to preserve bats in the face of white nose syndrome, if there's a way to help them not have to expend all that energy to go really, really far to get food, that can help them recuperate from infection and be able to survive to have young year after year. Yeah.
That's really cool.
Yeah, I'm excited. Not that I've ever driven a drone before, but we'll figure it out.
Yeah, I noticed on one of your graphics, you said the bat species 42% in Wyoming. What makes Wyoming so unique?
Gosh, yeah. Wyoming is a very unique place. I think it's because we have so much open habitat and such a diverse suite of habitat, where we range from rocky outcrops to a contiguous forest to sagebrush steppe and everything in between. So we are really interesting in that even at our latitude, we have a high number of bat species compared to other states kind of within this region. I mean, even Tennessee only had like 18 species. So we don't know exactly yet why it is that we have that. It could just be that some species pass through and they're moving from, up to Canada all the way down. But for some of these, like our cave myotis, that's that new species, or the canyon bat that Ellen Whittle detected, it could be a function of them using the landscape and the coming up the river corridors and just kind of happening to be up here. Yeah, it's a good question, and ultimately we don't really know. For our tricolored bats, as an example, we are the western part of their range, and we only find them hibernating here. We have no idea what habitat they use to get here and why they don't summer here. So we're trying to figure it out. If only I could just talk to them and ask them. But yeah, great question. I wish I knew answers for all of it.
So one thing you mentioned was that these bats need a vertical distance to drop down if they have a bat house in order to gain flight. What are some ways that folks can do things in their backyard or in their community to help support a healthy bat population? Do you have recommendations for people that want to help support bats?
Yeah, absolutely. So having nice forested habitat's always a good one, because bats do live in forests and rocky outcrops in our region. If you have a bat box, having it in a place that is high enough is good, and usually it's best if you don't have it on your house, because they'll often be like, this is great, but your house is better. So to avoid that kind of conflict. There's a lot of great plants that you can have that bloom at night that could enhance insect availability in your backyard. So they're kind of bat-friendly gardens is another way. And then of course, which is a little bit of a tough one, is that if you do have cats, to try to keep them indoors, because they do eat a lot of bats. That's one of the top contenders for small mammal and amphibian and herpetofauna that succumb. So leashes on cats are great, just like dogs. But those are kind of the main things. Yeah. Mm-hmm.
So there was a graph right near the beginning where it was about the trapping of the bats and how they've actually declined. Is that a loss of the species, or also they are moving to other locations due to climate change and weather as well?
Okay, so you're talking about one of the white-nosed ones? Things changing that way?
That was, yeah.
Okay. Yeah, so most bats have high site fidelity, meaning they come back to the same places to winter and to summer year after year after year. So it's kind of like ungulate, like the ungulate migrations we've seen in Wyoming, right? They follow the exact same route to their summer grounds back to their winter grounds. So bats kind of do something similar. And what's likely happening is that the decline in species that we're seeing is potentially population extirpation due to disease and other factors that are happening on the landscape. So we're seeing populations of these myota species completely go away, where we will never capture them again. Hopefully that's not the case, but so far it's been very challenging. And what happens then is that our myota species do not migrate very far. Like they overwinter here, they summer here. So if you're starting to see that massive decline in summer, that's reflective of what's happening in the winter. And then if you have a low number of individuals to mate, then you're gonna have low recruitment. And with 1 pup a year, that means we're gonna basically see a snuff out of some of these species. To counter that, we have an increase in some of the other ones. However, what's happening in climate change or on the landscape could affect those species as well. And so the fact that we're seeing a decline in our hoary bats is very concerning. They're on petition for listing, so they're on the U.S. Fish and Wildlife Service agenda for 2028 to see if there's something that we can do to minimize that or to get them listed. 2028 is a long ways away. And so with that means that we're trying to do as much research as we can to try to get at some of those questions to figure out, why are we seeing declines of these hoary bats? Is it just last winter was a fluke and it was just a cold winter with a long wet spring and the bats just weren't doing well and maybe this year they're doing better? Or is there other stuff going on around the habitat? So what Renee is doing is that she's going to be looking at her place base, so the National Park Service, but to try to get at that hoary bat question, she's going to be looking much broader at seeing what kind of habitat change has happened that could be leading to, so it might be kind of hand wavy, but what could be leading to declines in hoary bats or those other species that aren't getting hit with white nose syndrome. But yeah, it's not looking good for our bats out here. We're seeing complete eradication of our species in our caves too, based off of winter surveys. Yeah, good question.
Are there any, I mean, I'm sure there's research going on how to treat white nose, maybe how to clean, you know, if a cave gets eradicated, go in and clean it, or maybe it self-cleans over time. Vaccinations, what's happening on that front?
Yeah, that's a great question. So since about 2010, folks have really been working on a variety of different management or mitigation techniques and treatments. So there's been a lot that have been in like the research and development stage that's just we're not going to kind of get off the floor really. There are some things that folks are working on and that Wyoming is actually a part of, and a huge collaborative effort led by USGS, Wyoming Game and Fish, and then we have National Park Service, among others, that are contributing to the vaccine trials that are coming. And so we have actually a site, first time out here near you all, at a site by Yellowtail is going to be on the vaccination trials, and then Fort Laramie is this huge, massive colony of little brown bats. That's 7,000 individuals in this maternity roost where I'm going to be helping them next week vaccinate. Right now it's just at trial stage, so it's like capturing bats, putting the vaccination directly into their mouth and then releasing them. And so they're trying to see what is the efficacy of that. To a point to where we can actually just start smearing kind of a vaccine goo on some of these locations so that every time they crawl across it to get to their maternity roosts, they have it on their bodies, they groom it off, they groom off each other, and then you can actually have this vaccination happen. There's also some folks who have been working on creating bat buffets near cave sites where they are erecting lights near these cave sites to kind of bring all the insects in so that bats are leaving these caves, being able to fatten up but not have to fly too far. Because one thing that occurs with white nose syndrome is that because they're going through torpor arousal so frequently, they often are dying of starvation versus just the fungus alone. So there's been a couple things working on that, maintaining habitat, trying to make really nice habitat close to those roosts. And then, like I said, with the Y dot is that in these artificial roosts or these human-made structures, we don't have to worry about any non-target effects if we just go in there and flamethrow the whole thing, clean off any PD, and then the bats come to a clean site year after year. And so there's been some cool work in Tennessee where they created a whole artificial hibernation site next to a endangered gray bat cave. And that's a site that these bats can go into in the winter, and when they leave, they can completely just clean, absolutely sterilize that cave surface, that fake cave surface, so that the bats just come into this nice, clean location when they overwinter. So there's some pretty creative alternatives or management actions that folks are coming up with. But yeah, Wyoming's kind of some of the lead for this vaccination trial right now, which is pretty exciting.
We got time for one more question. If not, I have one. Please.
Does the PD fungus affect anything other than bats?
No, so I guess that's a good thing. No, it's so it's a fungus. It's a cold-loving fungus, and so it does fine in caves on its own. It's kind of a poor competitor. So if you were just to grab a soil sample from a cave, all this, and try to culture it, a bunch of other things are going to grow, but PD will be in there. But when it finds bats, it does really well. That's its food source. And so when bats go into these cave sites and they reduce their metabolism, they reduce their blood pressure, everything is just reduced. So there's still animation, and it has evolved to be able to infiltrate that mammalian tissue and start eating away at it. And once it does that, it gets this little cap on it and has a complete different physiology. And it's we have some folks working on some projects looking at PD at LCCC, Laramie County Community College, with Amy Wangline and Braden Mays. But it's a really good invasive species for bats. It's a really good disease, but it doesn't hurt anything else. We're not seeing anything like the raccoons or foxes that go into these caves come down with anything. It's kind of similar to salamander chytrid fungus or chytridiomycosis in general, where it is evolved to do really well on this one host. It has some hosts that survive because they have some sort of tolerance or immunity of some sort, so they're not completely killing off their host, but it's really host specific as far as that goes. You don't see ducks or moose that are walking through those ponds getting chytrid fungus. So fortunately for other critters, they're okay. For bats, not so much. Yeah.
Dr. Bernard, thank you so much for your time.
Yes, thank you.